Canary introduced us to the cultivation of yeast cells and how they could be used in our experiments. Our professor conveyed to us the importance of sanitization in a laboratory setting. This lecture included a short introduction to the autoclave machine and its amazing properties to sterilize all equipment. How we used the autoclave was by sterilizing micropipette tips, which then led to the introduction of micropipettes. Micropipettes are instruments used in science to accurately transport liquid in specific volumes. Lastly, we learned about shrinking and how we use it with the inoculation process, essentially teaching us the needed basics of a lab setting. We first started gathering our materials we needed for the experiment. Of course, the thing we need most of all is microplastics. To help with the production of microplastics, Ruben and Marisol were assigned this task. We then weighed them out in an Erlenmeyer flask and labeled them along with our mini petri dishes. We then started making our first inoculations. Ruben, Violet, and Marisol began scratching the yeast with a tip and adding it to the media samples by rubbing it around the edges. This will help us later with having enough yeast grown to use in our experiment. Dr. Canary showed us how to properly dispense and measure the microplastic. Skylar and Ruben have been working on measuring the microplastics that we have collected. While the others were busy, Aineen and Violet were researching for more information about the microplastics, how it affects the yeast, as well as updating our journal on a daily. We began measuring the microplastic by grams. After measuring, we would put the appropriate amount into the corresponding Erlenmeyer flask, putting 1 gram, 0.5 of a gram, and 0.1 of a gram. We used mini petri dishes from Dr. Canary. Before we could use them, we had to take the solution to be poured in there first. We mixed hot yepid and the microplastics inside like you see in the video. This causes a chemical reaction for the solution to crystallize, making it impossible to put into the petri dishes without moving the liquid around. We also started our overnight culture with them making a set of 3 by 3 of 1 gram, 0.5 of a gram, and 0.1 of a gram. We used some of the well plates. We got a set of 4 out of 3. Skylar and Ruben began adding the media into the well plates with some microplastics using a pipette. Once done making them, we took them to the lab for some tests, and of course we got our results. We largely used plate reader to get our results. We repeated this process over time, as well as repeat overnight cultures. Our results from the plate reader and the overnight cultures told us all that we needed to know. While we worked with yeast cells, Dr. Canary worked with human cells, which are more delicate and have different requirements. We came to the conclusion any percentage of microplastic would completely kill the cells. We also put the yeast cells in a Bradford assay to better understand how and why the yeast cells died. Here you can see the various steps we took to get the cells ready for the Bradford assay. Thanks to this, we now better understand how microplastics affect eukaryotic cells. Mr. Bailey, say hi.